Well, thank you very much, Magdalena, for your kind introduction, and thank you, Bengt, for your kind invitation. I'm enjoying very much this meeting. I'll talk today about a work in evolutionary bioinformatics in our lab. We are interested in understanding how structure plays a role in the evolutionary process. And so basically, if we are to look at the roots of life, there are two main approaches. The ones we've heard from Craig and Jack yesterday, where we use an approach of basically creating components based on information that is extant, that is present in today's living systems, and we try to basically reconstruct or build a living system from those components. However, an alternative approach, the one that we've focused on, has been to reconstruct systems. That means to utilize information from extant organisms, generate, reconstruct, or replay basically what ancient life would look like, and then with the hope that we could then apply this to build components in the living system. And this is what is called resurrecting ancient reconstructions. You can actually resurrect artificial entities that are supposed to be ancestors. So the basic premise of my talk is that structures, molecular structures, are living fossils. And these structures correspond to the polymers of life, proteins, nucleic acid, lipids, and many others, but notably these two, which are basically the machinery of the cell. And these are definitely much more complex than their string representations. So it is clear that as we travel from the Armstrong level all the way to 1,000 Armstrongs level, we are increasing in this complexity as we see unfolds the folds, the structural folds present in tertiary structure, quaternary structure arrangements, and even complex formations. So this is basically traveling from the sequence all the way into the cellular domain. And as we do that, what we find is that change occurs very rapidly at the sequence level. If a rough calculation of the numbers of changes that you would expect on Earth in terms of sequence, new sequence that covers sequence space, would be at the millisecond level. So every millisecond you will have many new sequences coming to play. But as you travel this way, towards molecular complexes, these structures take more and more time to be discovered. And here we require millions of years, if not billions of years, to generate some of these very complicated architectures. So that means that as we travel this way, evolutionary conservation increases with this hierarchical complexity. One aspect that is also interesting that I want to point out is that here we have orders of magnitude above what would be expected from a gene duplication. So it means that these structures accumulate during millions of years in the proteomes. Therefore, we have a high representation of these structures with time. One aspect that is important also that I want to pinpoint is that the macromolecular world is modular. It is built from pieces. And a couple of examples here in proteins, the supersecondary motifs, like for instance these helical structures that appear recurrently in individual molecules. And in RNA, we have also helical segments that are recurrent, that you see appear in complicated structures like the ones you would find in the small subunit of ribosomal RNA. this modularity is also evident in the actual complexity of proteins. These proteins are built from these domains. These structures are highly compact that are recurrent in different proteins, in different environments, and establishing a complex architecture where you have individual structures of well-packed helices and stems and other elements, and that organize into possible domain combinations in multi-domain proteins. So you have a modular arrangement of these domains in proteins that also show that modularity of the protein world. So, so an effort to visualize this complexity has been to create classifications of proteins. Like, for instance, the structural classifications of proteins showcases this hierarchical structure that I was explaining about, where you can basically pool on the top of the hierarchy the groups of classes, and then from here all the way to fold structure, fold superfamilies that are highly conserved and evolutionary units, all the way to fold families, and ultimately the domains. Other classifications like Cath have alternative classifications of the protein world that allow you to fine-grain this complexity in a better way. And here you have kind of the idea behind this classification. So now, in our lab, when we've looked at domain diversity in proteomes, and when we look at large numbers of proteomes, let's say of free living organisms, it becomes evident that the diversity of domains that a proteome can have, the occurrence of domains, versus the domain reuse, the abundance of the domain, are important and somehow already dissect the diversity of our living world. Like, for instance, eukarya has incredible levels of redundance, reuse, at the same time having incredible levels of diversity, while archaea is on the other border of the spectrum. And as we travel from here all the way up to Homo sapiens, you know, we are increasing both aspects of that equation, meaning that, reuse and diversity are somehow linked to complexity in life. So we've elaborated on this, and we try to do this from a topological perspective in terms of structure. But the problem here is that there's no metric that allows to compare structures to each other at a global level. And so a way to overcome this has been proposed by Bill Taylor in the UK, where he would try to generate a periodic table of structures that would somehow distill the complexity of the structural world. Of course, by having this table, now you need rules to describe how these structures change from one to another. And this is the aspect that is very difficult because we don't have those rules. So we bypass this approach by going into an evolutionary view where we use abundance as our criterion to determine shared and derived features at a phylogenetic level. So we basically use the standard tools of phylogenetic analysis to reconstruct trees, very much like the Darwinian trees, that will describe the evolution of the protein world. And we did this in a number of publications. I've showcased some of them there. Let me explain the methodologies. It's relatively simple. Given proteomes, we can assign structures to sequences on the entire complexity of the proteomes using hidden Markov models that recognize the structures that are known and basically generate a census, a genomic census of structures, where we take each and every fold conformation and we count how many of these fold conformations exist in a proteome, let's say Bacillus subtilis. And this will be represented by a column in this gigantic matrix of ever-expanding genomes that are being sequenced in the order of the thousands. So by doing this, with this matrix, this is a very valuable piece of information that gets updated as new structures are added and as new genomes are added and a new classification scheme is revisited in time. So this has been evolving during the years in my lab. And what we find is that given this data matrix, we can now reconstruct 3 kinds of structures, and I'll showcase those. First of all, a tree of the system, that is the proteome of the organism. And in this case, and here the player is Menglei Wang in my laboratory. I've been, updating all these reconstructions, every few months. And when you reconstruct this tree of proteomes, the leaves of the trees are basically the proteomes, the organisms. And we can reconstruct trees that really show the 3 domains of life: archaea, eukarya, bacteria. Within the eukarya, groups that are well known are grouped together. And we find, interestingly, an origin of these trees, because these trees are rooted because of the principle of abundance. We find that the origin is in archaea, and we find and have defined a complex and universal cellular ancestor, a LUCA, as it's usually termed. I'll come back in a minute on this aspect. An alternative to this is to build this kind of trees, trees of architectures. In this case, the leaves of the trees are the component parts of the systems. That means these are the actual structures of the domains. And the tree is global in that it represents the globality of the protein world, and it can be reconstructed at different levels of the hierarchy. I showed it in that initial slide. These evolutionary patterns provide origin and structural diversification of this protein world, and we find that the origin of these reconstructions are in enzymes related to metabolism. So the third approach, and this has been pioneered by Kyung Mo Kim also in my lab, has been to use the link between structure and function, and just use the gene ontologies, like the one that is published in Gene Ontology database, to make consensus and generate now a phylogenomic tree of functions, where the leaves of the trees are basically the gene ontology classifications. And to our surprise, we find that beautifully, again, the metabolic functions are located at the base of these rooted trees. The interesting aspect, too, is that biosynthetic processes, very much like the ones that you find in the production of proteins, are derived, are latecomers in evolution. So these are definitely 3 kinds of trees that then you can map, for instance, on networks. That means that you can basically take the information of age that is provided by these trees and map the age with nice colors, red meaning very ancient, onto metabolic networks. And what you see is this patchy structure, almost a mosaic structure, that tells us that networks basically grow by recruitment. That means that they are different structures and functions are being taken from one place to another into different contexts in a very haphazard manner. So we are very interested in recruitment as a powerful force and an unanticipated force in evolution, and we are trying to sort out these processes. The other thing that we've done, we've traced the age of these domains that come from these trees onto molecular complexes. And this is the focus of my talk. Basically, we are interested in unrooting the biochemistry of life, the origin of life, by basically tracing the age of structures linked to proteins and RNA into these complexes. So this is work from my son, Derek, in our lab. And he has basically taken the tree representation that describes the component parts, the evolution of the component parts, and turned that tree representation into a timeline, where 0 represents the origin of proteins and 1 represents the proteins of today. So what is fascinating about this is that at the beginning, you have all these gray pie charts. That means that these are structures that are common to all the living world. It means that at the beginning, proteins were common, as you would expect. between all organisms. Then you reach this epoch 2, this superkingdom specification, when all of a sudden we have a new class appearing of structures or domains that are shared by eukarya and bacteria. This is signaling that perhaps there is an increase in reductive forces in the evolutionary equation. And I'll come back to later on. And then later on in epoch 3, the organismal diversification epoch, we start seeing all these complex arrangements of folds that are specific to the different domains of life, specific to archaea, to bacteria, eukarya, or to combinations of them. Interestingly, there's an hourglass pattern appearing here. And this hourglass pattern we've seen with functions and with many other aspects as we trace them in these evolutionary timelines, and we're trying to figure out why you would have this increase, this peak occurring first in the architectural diversification and then in organismal diversification with a constraint in this region. So there's an hourglass pattern that we need to explain. Now, one aspect that is interesting is that if you use geochemical evidence and you use fossil record to basically associate those aspects to structures, then you can somehow generate a molecular clock. That means that we can now map the age of the protein folds that come from the individual trees that we've been reconstructing down to the actual geological timescales. And to our amaze, we find there's a very nice straight line, nice correlation that shows that we can basically time events that we can derive from the trees into this geological timescale. So we've done this, and in order to prove the concept, we basically explore metabolism and determine all enzymes that were aerobic, anaerobic, or that have both features. And we find out that aerobic metabolism basically comes 4 million years before the great oxidation event that has been very well timed by geochemical and isotopic fractionations and many other techniques. So this is interesting. And we also studied the makeup of the last universal common ancestor by basically studying what is the minimum, the repertoire that these trees unfolded that was common to all life. And we find out that LUCA basically appeared during the time of that great oxygenation. So that means that diversification somehow coincided with the rise of oxygen in our planet, which is, I think, an interesting combination. So we basically, in my talk, will try to map all these events that are important to this timeline and see whether it makes sense to find where they appear. So one thing that we find out some time ago is that the majority of functions, as mapped by the Enzyme Commission classification of enzymes, the majority of those functions appear in our timeline at the beginning of the protein world. That means that there was an explosive rise of function in the makeup of this primordial metabolism that basically encased the majority of all modern functions. And this arises very early. So this is what we call this metabolic big bang that occurred very early. The other thing that we find, which is very interesting, is that when we plot in those timelines, we plot the number of, or the fraction of proteomes that contain a particular domain architecture, whether that be a fold or a fold superfamily, like in this case folds and fold superfamilies, we find that there are clear decreases in the representation of the individual architectures in the proteomes that have been sequenced. And first we find a marked drop occurring first in archaea, then in bacteria, and then in eukarya. And so this is signaling that there have been reductive evolutionary tendencies setting up in the world, first in archaea, very early in evolution, prior to the LUCA, then in bacteria, and finally in eukarya. Interestingly, however, the folds specific to archaea and eukarya appear together at the time when eukaryote-specific folds appear in the timeline, quite late in this timeline. So this is interesting, and we've followed by now reconstructing trees of proteomes, but doing it in a very particular way. So given a tree of domains, we can separate the structures, the protein structures that are very ancient, the ones that appear during organismal diversification, and when the domains of life were very well set, and reconstructed trees in those three cases. And to our amazement, we find out that the trees reconstructed from very ancient structures were all rooted paraphyletically, that means within the superkingdom, in archaea, and the same occurred in this intermediate phase. But then when we would reconstruct using recent architectures, we would have the canonical tree, the one that Carl Woese put forth, where you would have a rooting of the tree in bacteria, with archaea and eukarya being derived. So this is telling us that different portions of the world of proteins is providing differential signatures to the history of the living world from the system perspective, but also from the component part perspective. So, so basically, this is our work and hypothesis. We have a period where we had structural change occurring. This is cells' concern about discovering new properties in the cellular domain. Then a communal phase, and not referring to the communal ancestor of Carl Woese, but a different communal phase, where you would have these reductive tendencies in archaea setting up already in the protein world. And then an organismal phase that basically matches what sequence have been provided us in terms of information. So this has been all nice, and we basically characterized the organismal diversity from the structural point of view, but we were interested also in understanding this modularity that exists in the protein world. So some time ago, we published these trees that are very peculiar. These trees are not only domains, structures, but these are domains and domain combinations. So it means that we are describing here the modularity or makeup of the individual proteins. So each of those leaves represents either a domain or a domain combination. And what we find is that quite late in evolution, there is a big bang in the protein world where these combinations start occurring and producing these multi-domain proteins. And this is evident, you see here in domain pairs or multi-domains, versus single domain growth that seems to be relatively constant in terms of architectures and or fold architectures or fold superfamily architectures. So now Faiz Aziz in the lab has done a simulation of how this combinatorial game pans out, and what I want to show is this movie, if I can. Oh, there it is. So what I'm going to show, what I want to show is how these connections between the individual domains form in the protein world. And you're going to see, starting from 0, that means the beginning of the protein world, all the way to the present, when you'll see that turn into 1. And what you're going to find out is that at the beginning, there's not much occurring. There are some few connections of proteins, domains, that are somehow linked to each other. But as time progresses, this basically increases, that intensity of connectivity increases. And as you can see, those dots, which are the domains or domain combinations that share those arrangements, appear larger and larger. That means that there are donors, basically, in this. And all of a sudden, the Big Bang occurs. So this is the Big Bang of domain combinations. And look at this. You see these black dots. These are donor structures that are being delivered, and reuse and reuse and reuse over and over by genomes, all the way as we are approaching the present. And as you can see, this is heterogeneously, provided to a number of new architectures that are being generated with time. So this is, I think this visual representation shows this phenomena of modularity setting up in the in the protein world, which is definitely of interest. So what we've done is basically go family by family, like for instance the scaffolding proteins, which we know are latecomers, important in multicellularity, and we've mapped all the domains that are linked to this 100 and plus structures, and found out that they are basically appearing when they should, after the appearance of the eukarya and during the protein, the Big Bang of the protein world, followed obviously by cell adhesion receptors, proteins linked to multicellularity. So we mapped. There is incredible information in these trees because each of those architectures are linked to a number of functions. So depending on how you map this, you'll have a lot of information about evolutionary progression. So one aspect that we've done also, which is of interest, is the evolutionary mechanics of the protein world. So that means that if in the timeline we had a domain that at some point appeared early as a single domain, and this domain combines with another one later on, that appeared later on, to form what we term a fusion combination. From fusional domains, we turn them into fusional combination. But you could have different schemes. You could have a fusional domain interacting with another domain in a combination at a later time, which we call fusional-fissional combination. And then this one will excise a new domain, D, in a fissional, and this is a fissional domain later on. So you have different alternatives, and you can map those alternatives in the timeline. So we found out, amazing, that there is a set of domains that do not combine with each other, that appears very early and also during the Big Bang. But we found that fusion seems to dominate the early stages of the protein world, and fission seems to dominate, especially in the eukarya, the later stages of evolution. So we've basically mapped some of these phenomena and mapped the structures and the functions linked to the fusionals and fissions. And we find out that in eukarya in particular, like here in humans on Thaliana and Arabidopsis, we find that there is a large number of fissional proteins that appear very late in evolution and that seem to be crucial for a number of modern functions in this genomes. So now we've explored what are the primal or fundamental events in this timeline, and mapped events linked to the ribosome, linked to DNA replication, linked to the appearance of elongation factors, the appearance of aminoacyl tRNA synthetases, and we find out that in all cases, the domains that appear first in the timeline are all related to catalytic functions within these roles. Like for instance, elongation factors and aminoacyl tRNA synthetases, which are the first that appear in the timeline, linked to translation, are all, the birth of them appear in this red ancient structures that are catalytic, the catalytic domains that process that energy needed for their functions. And that the areas of these molecules that interact with transfer RNA in those cases, it's a latecomer. So this is against this idea that RNA could be so ancient. It appears that appears later in the timeline. So the question is, can we map RNA in the timeline, RNA-protein interactions in the timeline, and somehow try to dissect the existence of RNA world, a protein world, or a ribonuclear protein world? And so we've done this, and for that we focus on RNA, and we try to link the timelines of proteins, of RNA and proteins, by looking at the structure of RNA. And we've done this with, again, a cladistic approach, an approach based on phylogenetic analysis that has a character coding step that establishes features that are molecular, and then assigns numerical values to those features. And we have then a phase of character augmentation where we put forth a model of evolution, and then a phylogenetic reconstruction. And look at this, which is interesting, this diagram, from these data matrices that are built from statistical and geometrical features in RNA, we build trees of molecules of the systems and trees of components, the substructures that exist in the system. And we've done this with a number of molecules, and here is just a description of how we use the statistical features that describe uniqueness, stability, and branching of the different RNA conformations, or geometrical features that describe the actual geometry and branching of the individual RNA molecules. And we use this information basically to generate these data matrices and a hypothesis that that is based on considerable theoretical and experimental evidence where it seems there is an evolutionary tendency to diminish frustration and increase order in molecules as evolution proceeds. So using this, we've basically explored a number of phenomena that go all the way from years to billions of years. And in all cases, we have a good match between sequence analysis and structure analysis in this phylogenies. With this phylogenies, we can color molecules according to their age, very much as what we've done with proteins. So the reddish colors are very ancient helical segments in, let's say, 5S ribosomal RNA or RNase-P, and the bluish colors are very recent. And we've done this with a number of molecules. I'll just show transfer RNA and the ribosomal RNA as examples. So there have been some influential hypotheses in terms of the evolution of transfer RNA. For instance, the fact that the top half of the molecule is ancient and was part of, involved in acting as genomic tags during a very primordial phase, and that this segment hosted an operational code, a primitive genetic code that preceded the modern code. So we basically confirm this distinction, and we've seen that stems that are related to the acceptor arm of the transfer RNA molecule are at the base of the phylogenies, and the bottom half is derived, as you can see. So, and we've done this with stems, but we basically explored hairpins, unpaired regions, other characteristics linked to the geometry of structures. We've done this also using statistical characters. And the interesting thing is that these trees allow you to put a model of molecular evolution, an explicit model of molecular evolution. So the tree basically describes a flow diagram of this kind, where you show the protostructure basically a build-up of the individual components as they are being added in evolution to the molecule, forming first, a ancestral half and then a derived half of that molecule. Interestingly, there are two pathways. One that is specific to bacteria and eukarya, and another one that is specific to archaea, which is also indicating perhaps a link to this reductive tendency that we've seen in archaea at the proteome level. We've done the same thing, and this is the work of Ajit Harish that followed the beautiful work that Fengye Sun has done in terms of this bioinformatic analysis. Ajit basically exploited the information that exists in the European ribosomal RNA databases on the small and large subunit, thousands of sequences, and he built this beautiful tree here that describes basically the evolution of the component parts of the ribosome. And as you can see, the most ancient part of the ribosome is the ratchet, the ribosomal ratchet that allows for translocation to occur in the molecule. And the actual PTC, the peptidyl transferase center, the responsible for protein synthesis, is quite derived. It's this yellow portion that you see here in the tree. And I want to note also this interesting thing. There is a split of two segments of this peptidyl transferase center. And this coincides with the proposal by Ada Yonath that there was a split or duplication that was responsible for the makeup of the protein biosynthetic site in the ribosome. So this matches that information. This tree matches that information, but it places the origin of proteins in a quite derived aspect of the ribosomal structure. So this is the three-dimensional view of mapping the age of those structures. Again, very ancient ratchet component here in the small subunit. Some structures that are ancient are related to GTPase center of the large subunit. And in yellow here, you can see the peptidyl transferase center being quite derived in the timeline. So this, the origin of the ribosome, according to these results, show that it's in the processivity center of the ribosome that is responsible for messenger RNA helicase activity, mRNA decoding, transfer RNA translocation. And so this is the core of those functions, and those delimit the origin of this molecule. But while this is very interesting, the most exciting finding is where we mapped the actual age of the ribosomal proteins onto the ribosomal ensemble. And we find out that there was a very nice correlation between the age of the ribosomal RNA helices and the age of the ribosomal proteins. So that means that the ribosome, the proteins and RNA co-evolved from the beginning of this interaction. That started with the most ancient ribosomal proteins, which are S12 and S17, both of them having the OB fold, barrel structure. And this continued all the way until the L7, L12 protein complex that stimulates GTPase activity appears in evolution. So this basically slide summarizes that progression in evolution from the processivity center all the way to this stage where the two subunits come together to form a operating system that now has the PTC, the peptidyl transferase center, and then this proto-ribosome basically continues to grow all the way to its mature form. And so this is this transition that we name that appeared around 0.3, and this is roughly 3 billion years ago, according to our timeline, and that brought the two subunits together to basically produce this event of protein synthesis. So just getting close to the end, so we've mapped the events before ribosomal protein synthesis. So how was the protein machinery built before the ribosome was fully functional? So that was our question. So for that purpose, we focus on the 52 fold family structures linked to the origins of the protein world. So that means here we have the first ribosomal proteins appear, and we wanted to know what happened here. What we found is that the majority of these structures were built on a layered architecture. This is again Bill Taylor's periodic table, and the origin of the protein world seems to be based on this kind of structures. The 3-1 design. That means layers of sheets spanning alpha helical components. And this seems to be a primordial design until barrel structures appear somewhere, very recently when the non-ribosomal protein synthesis machinery appears in evolution. So before ribosomal proteins, there is a number of structures that are crucial that we've mapped in these very early times into the timeline. And first of all, we find that the origin of proteins is in ATPases and AAA ATPase mechanoenzymes that all harbor the P-loop hydrolase fold. So this is a very unique design that interacts with ATP, has this ATP binding site, the P-loop. But very curiously, these ATPases and AAA ATPase mechanoenzymes have this helical bundle that is relegated to a part of the structure. This is quite unique within the P-loop hydrolases. And we make a lot of fuss about this aspect there, especially because when you split the structures into smaller pieces like Cath, the classification does. We've used SCOP all this time. But if you split it in smaller pieces and reconstruct trees, we find out that those trees, the most ancient two structures, are basically this helical bundle and this layered sandwich structure that appears in evolution. So So we see a recurrent appearance of first these metabolic enzymes, then followed by elongation factors, by aminoacyl tRNA synthetases, and then non-ribosomal protein synthetic modules. And given this progression, we now postulate that the evolution of protein synthesis proceeded first by developing this two-step reaction, aminoacylation reaction, that turns basically an amino acids into primordial peptides, and that uses class 1 and class 2 aminoacyl tRNA transfer synthetase enzymes for that purpose, which very recently have been discovered have the ability to produce the peptide reaction, but produce small little polypeptides. So later on in the timeline, we find the appearance of fatty acid synthesis, and then later on, non-ribosomal protein synthesis, all before the appearance of the first ribosomal proteins and the ribosomal machinery that we map in the timeline. So, so it is this layered design that basically supports the early evolution of proteins in the protein world. Now, interestingly, the first two architectures that appear in evolution are linked to membranes. These are the ATPase-like transporters, and then the AAA ATPase are chaperones that usually associate with each other in performing crucial functions. Some of them modern, but some of them very ancient. And we think that these chaperones have the ability to act as chaperones to incorporate ectopically small polypeptide and membranes, and then grow these small, bacterial-like peptides as membranes develop in time into much more complicated bundles, very similar to the bundle you will find in the ancient P-loop hydrolase folds. And then as time progresses, bulky extrusions of these primordial structures will become globular, and these will be the first oxidoreductase enzymes that you would have. And then later on in evolution, as new and more sophisticated bundles were discovered that appear quite late, by the way, in our timelines, then you would have the more mature ABC transporters. So this is kind of a flow diagram that shows an origin of proteins embedded in membranes being plausible alternatives. So basically, this is to summarize. We see a number of patterns related to architectural diversification, including a co-evolving ribonucleoprotein world. We also see modularity span out in the proteomic repertoires with an origin of modern biochemistry in membranes, perhaps, and layered protein topologies with the capacity to aminoacylate cofactors in RNA and synthesize polypeptides. These are, of course, the team and external collaborations: Chuck Kurland, Chris Dupont from Craig Venter's Institute, Frauke Grete and the Max Planck, Hongyu Zhang in China, and now Patrick Forterre, because we are very interested in origin of viruses. All this has been done with basically the cellular world. So what do viruses have to contribute in terms of structures? And this could be very interesting. And of course, we want to know those rules that dictate structural change in macromolecules so that we can complete Bill Taylor's periodic table. So this is just to thank you for your time, and an interesting quotation from Fritz Lippen in 1971. So thank you very much.